Hey everybody, welcome to yet another Journal Club here over at lifespan.io. I'm Oliver Medvedik, broadcasting from New York City. We're doing it a little bit, a little bit out of schedule. We're doing it Friday, but that's okay. Thanks for everybody joining us this wonderful Friday afternoon here in New York City and whatever time zone you happen to be right now. We have a journal article today. It's kind of a short article, but apropos to everything. It's related to aging research and related a bit to a topic that we might be discussing next month with a researcher who's well known with parabiosis studies, Irina Comboy. But this is not a Comboy paper. This is a different paper, which the authors here hope to isolate factors that are involved in tissue rejuvenation in blood. So let me share my screen here. Let's pull up this paper here that I believe you all have access to. There we go. I gotta move my. All right. Rejuvenating effects of young extracellular vesicles in aged rats and in cellular models of human senescence. So this is I don't know how to pronounce this. Shamagian et al. Paper. It's a Nature paper, Science Reports paper. And what they're attempting to do here is, well, a little background. Parabiosis is this series of experiments where you basically patch the circulatory systems between two organisms, usually with mice. They don't do this with humans usually, but they have been doing blood transfusions and a variety of other transfusions involving, I believe the last paper we covered was a Conboy et al. Paper discussing the dilution of blood with purified serum albumin and saline. And there were some noticeable improvements in health, some health span effects. And here in this paper, what they're attempting to do is kind of further isolate what can possibly be, one of the factors involved in transferring an improvement in health span. So when you do these heterochronic parabiosis experiments, you're tying in essentially, the first experiments were essentially patching in the circulatory systems of a young organism with an old organism, and you get rejuvenatory, if that's a word, rejuvenation effects in the older organism. But there's a lot of factors in blood, right? There's serum, there's cells, various types of white blood cells, red blood cells, and of course serum albumin, but all sorts of other things. And some of these other things are these, these components called extracellular vesicles, which are essentially small particles that are, lipid encapsulated. We've basically looked at some papers where people have looked at these extracellular vesicles that are shed or secreted by cells. They vary in size, I think, to the submicron range. Can't remember what the cutoff is in nanometers, but there's a lot of biologic molecules that are packaged in these extracellular vesicles. There's messenger RNAs, there's proteins, so there's still a lot of things in these EVs or extracellular vesicles. But be that as it may, this paper attempted to look at blood fractions and see, are EVs responsible for some of these health benefits? And, the punchline is, perhaps, maybe, yes. So what do they do here? So this is a paper that, primarily uses rats, and they use aged rats and also neonatal blood that's fractionated into 4 components, as they mention here: whole blood, serum, EV depleted serum, and purified extracellular vesicles. And they use this, and they use these vesicles to treat living rats, but also added these fractions to, as they mention here, old human cardiac stromal cells, CSPCs. I believe that stands for cardiac stromal progenitor cells. I was wondering where the P came in from that acronym. And so with these aged mice, they were given injections of these purified extracellular vesicles into, so these vesicles that they derived. So here's just make this a little bit smaller here so I can see this. Tell me how that looks. Does that still translate well? Can you see that?
It's readable.
Okay, yeah, it's readable. Okay, so they used basically neonatal cardiac tissue. They got these explants, and they derived these cardiosphere-derived cells. So this is basically clumps of, cardiac tissue that's growing in little clusters. And I don't know the exact protocol, but they managed to get these cells, these clusters of cells, to secrete these extracellular vesicles, and they did a purification of these EVs, and that's somewhere in the materials and methods. So I don't know exactly. I know some of the earlier papers we looked at, you can fractionate these into different sizes. So the EVs do come in different sizes. So I did a little bit of preliminary reading on EVs, and there's kind of a, so when you say extracellular vesicles, they come in different types. Some of them are secreted outside, packaged within a vesicle. So you can imagine vesicles within a vesicle that are exocytosed. And then you have others that basically are, much bigger and sort of bleb outwards. So I don't know exactly what types. So there's a range of these EVs, and again, I don't know what the particular contents of these EVs are. So anyway, despite that, they use these CDC EVs that were derived from neonatal tissue, and they had this protocol here where they injected, I believe, kind of around. Well, it was in the heart. I believe it was in, let's see, what area of the heart? I forgot. It was, I want to say it was by the, I think it was in the ventricular zone, but I'm trying to get the figure here. Intra-left ventricular injection, percutaneous. So what is that? Slightly underneath or at the kind of upper tissue layer of intra-left ventricular injection. So this was done in the heart, and so around the heart, directly around the ventricular area, right, which is sort of the dense, the denser lower part of the heart. And then they looked at essentially the areas of the heart, and looked at essentially what, there was any improvements between either PBS control injection or these CDC EVs. Now, they do a number of different things, suggesting that there's an improvement. The one thing that they don't do, and I'm just going to mention here right off the bat, is, we discuss, a lot of ways how you can determine whether or not biologic aging is improved. And unless it was in supplemental, which I don't even think it was in the supplemental here, I don't think they looked at, just to cut off anybody at the pass who asked this question, I don't think they looked at, methylation clocks or any other types of clocks that we've been talking about as far as an improvement. So there's an area to, area for experiments here, further experiments to determine whether or not actually, the methylation status and the methylation clocks have improved biologic age. What they did look at is they looked at telomere length. So this is in figure B. They looked at gamma H2AX staining, which is an indicator of DNA damage. And I believe this was after injection, how many days after that was? I'm going to say, how many weeks was this? So these are Fisher 344 rats. I'm not familiar with those rats. If anybody else is familiar with this rat strain, please chime in. Let's see. X amount of days, however many days that was. 22-month-old F3 44 mice, which seem like pretty old mice. Yeah, there were some weeks afterwards. I can't recall at this moment here how many weeks afterwards. You have to take a look at the materials and methods. But anyway, the bottom line is that there was an improvement in these two factors. So essentially they say that, staining for telomeric loci, the spot intensity, which is indicative of, so this is, I believe, fluorescence in situ hybridization, so fluorescence labeling of telomere lengths in this cardiac tissue was increased. Also less DNA damage. And I believe they also saw that there was an increased expression of nanog and other factors which are indicative of some reprogramming. And that was primarily the take-home message of the first figure. I'm going to, as we get into the later figures, I'm going to just kind of raise another issue here. As they look at other systemic improvements, one thing that's still not clear to me is that you have these improvements, and the improvements are, one of the main improvements, and I'm going to kind of go into figure 2, is they're looking obviously at the heart. So these are, extracellular vesicles derived from the heart and then injected in the heart area, so the ventricular area. Scroll. And they have, this is good. They have improved cardiac function. So, they first start looking at, whether or not you have further improvements in kind of the morphology and the histology and the physiology of the heart. And you do. So red is PVS, blue is CDC-EV, so 16 weeks in this case. So here's the timeline. So let's see, LV, change in left ventricular mass, change in, what is that? I've already forgot these acronyms. It's been a while since I read this paper. Okay, decreased thickness of the anterior and posterior left ventricular walls, and reduced left ventricular mass. So that's indicative of a healthier heart. So B is, what is that, a cross-section of, I want to say an artery, but it's probably a cross-section of the ventricular area. Mid-heart sections from a rat, so less thickening. Believe they have increased blood pressure. They have Mason's trichome staining, which is indicative of less fibrosis in the heart, so area of fibrosis goes down. So general improvement of heart histology and heart function. And regarding heart function, I think they do some cardioechograms where they basically, again, I'm not an expert in cardiology, but this is indicative. So this is a measure of elasticity. This is delta E over E prime. So the elasticity, I believe, is improved in these hearts. The function is improved after 16 weeks. What else do we have here? So an overall improvement in cardiac efficiency, which is good because you're using extracellular vesicles from the heart. And does anyone have a link to the paper handy? Oh, there's the link. So is there anybody here who has any questions about or has any insights into cardiac efficiency or anything cardiac related that they'd like to chime in with?
Yeah, I was noticing that the untreated heart got thicker and less efficient, and the treated heart was more like a young heart. Mm-hmm. I think that's an important structural factor.
Yes.
When they're thicker, they don't refill as rapidly, and they don't pump as efficiently.
Right, and I think the thickness primarily has to do with it's fibrotic tissue, right? It's or is it both cardiac muscle? I mean, it's certainly when there's damage to the heart, it tends to be replaced with less functional cardiac muscle, correct?
I think it is fibrotic because the old experiments with ALT 711, the cross-link breaker, also were beneficial in that factor. So I'm thinking that it's fibrotic related to glycation cross-links. Mm.
So that's a good question. I mean, I don't know if they could look at improved breakage of cross-linking. If there's any other experiments they could do to test that. So after you have an improvement of cardiac tissue, so one thing to mention is this is being injected into the heart area, these extracellular vesicles. and you don't only have improvements in cardiac function, but they say that the health span is increased. So this is delta distance. I don't know exactly what this is a delta distance of. It's that changes in maximal exercise capacity. So I don't know if there's like a little treadmill they run on, but that being said, that's improved over time, and the survival rate is improved as well. Follow-up days after treatment. So there's a threefold increase in latency to death. So there's an improvement in these mice, from dying essentially. So it's probably the best measure right there. But they also have systemic improvements in skeletal muscle, lung, so sclerotic glomeruli, so this is kidney function, so basically less fibrosis in a variety of tissues. The question I have, of course, is, if you improve cardiac function, then your entire vasculature is, you basically have better blood flow, you have, better, you have better systemic effects, but stemming from improved heart function, right? So I think a further question I would have is these EVs, if you're injecting them into this pericardial area, I don't know how many of them can get, move around and target other organs. So are all these systemic effects, and I don't think they addressed that in this paper, but are all these systemic effects primarily through an improvement of cardiac function? Or are these systemic effects partly also due to extracellular vesicles directly targeting different organs as well? And yeah, so I don't know if anybody has a thought or comment on that, but that was one thing that came across my mind. You would think that the most direct answer would be that it would be directly due to an improvement in cardiac, but you can do follow-up experiments with that and see if you can get extracellular vesicles from a similar system that's from liver or kidney cells, and see whether or not those EVs can improve cardiac function, or those EVs will be specific just to that organ. Thoughts, comments, anybody?
I think more experiments need to be done. I don't think we have an answer yet.
Yeah.
But I note that on page 6 at the bottom, it says EVs are the primary blood-borne rejuvenating messengers. And so their speculation is that perhaps some of the other effects of young plasma might be due to the EVs in the young plasma. But I think that needs to be tested.
Yeah, and they do kind of the more like telling well, those are all good experiments up there, but they go further where this is kind of a cross-species effect here. I believe in this next panel, they use different fractions of EVs, and these EVs are main chemistry in neonatal serum. These EVs, correct me if I'm wrong, these EVs are purified from. So these cells. So this is a tissue assay that's from humans. Where does it say here? 55-year-old. Yeah, 55-year-old human donors. Actually, main characters heterochronic. And these are basically from neonatal rat blood. So this is a cross-species effect. So they've got these EVs isolated from rats, right? So neonatal rats, and they've got a whole blood serum, depleted serum EVs, right? And they test the various fractions on similar cardiac stromal progenitor cells obtained from over 55-year-old human donors. And they're only looking at, again, gamma H2AX, DAPI staining, Ki67, which I believe is a marker for replication, and beta gal, which is a senescence marker. And so basically in this panel here, which is all blood, S for all serum, D is depleted, meaning that it's got EVs removed from it, and then purified EVs. Basically what this suggests is these beneficial effects. If you just have EVs and you isolate them from all the other blood components, that you have a similar effect to if you have serum containing EVs or whole blood, versus having a depletion, which basically has a much worse effect. So this is interesting in that these effects, whatever this component is, seems to be very similar across mammalian species, at least between humans and rats. The other question I have is if anybody recalls the experiments that we did a paper a while ago with it was a paper from the Comboy lab where we had an improvement in health span in serum that was basically serum albumin that was removed from serum and then resuspended in saline. So you had essentially a isotonic solution, so equivalent concentration of salts to blood, and that was used in a plasma dilution study. And you had I can't recall all the health effects, but you had an improvement in a variety of parameters here. And I don't know if the Comboy paper looked at whether or not when the serum that they purified and resuspended with albumin had extracellular vesicles in there. They were just focusing on a purification of serum. I don't know the actual protocol for purifying serum from blood and whether EVs tend to come out of there, but they had beneficial effects. So in this panel here, they would if you're having this depletion of EVs, you would see it would seem to suggest that if you're having these negative effects from depleted serum of EVs, and this is neonatal serum, and yet they're suggesting that they have senescence go up. I'm just trying to figure out how we can reconcile those two results here.
There's an important difference with the Convoy experiment.
Mm-hmm.
They were donating it to live rodents,
mm-hmm.
and in this experiment, we're looking at cells in a petri dish.
Yes,
and we know that if you are a human or a rodent and you're diluting your plasma, that your liver is making new plasma within a couple of days. And in a petri dish, that's not happening. So that might be an important difference.
Mm-hmm. So yeah, that is a key difference. Just wondering if there's any additional differences. It would be interesting to see if, related to what I said earlier about EVs having a direct effect on tissue types, whether or not, since you have this, I'm going to just stop sharing here. They have this assay here to test whether or not EVs are functional, and you can see if certain parameters are improved, and maybe throw in additional parameters like methylation clocks, whether or not they can test these EVs on different tissue types, right? Because this is still a similar tissue types. It's cardiac cells, cardiac stromal cells, albeit in human cardiac stromal cells. But would we see these similar rejuvenatory effects, EVs derived from neonatal cardiac cells? Would they have a similar effect on hepatocytes? Would they have a similar effect on fibroblasts? Would they have a similar effect, or are these EVs more specific for a cell type?
That's a very good question, and it raises the question: why did they decide to start with this particular cell type?
Yeah, they did an earlier study, but again, I'm not sure if that actually would address this particular question. Yeah, I didn't read that earlier study that they did. But I don't think in that study they were narrowing down to see whether or not these EVs would have an effect on particular cell types. Because as I mentioned before, obviously improving cardiac function would have wide-ranging systemic effects. Brain, everything. So is it the heart being specifically targeted here, or are there other wide-ranging effects, or is there some kind of specificity, or are there categories of EVs that can have more kind of wide-ranging effects on cells? I don't know. It's a good question. Further experiments need to be done. But it is intriguing that these extracellular vesicles do have this capability. And we looked at it earlier, it was some journal clubs ago, where they used purified EVs to basically rejuvenate skin. And they were looking at skin function. And I can't remember right now, I can't recall off the top of my head if those extracellular vesicles were derived specifically from skin cells, or they were also looking at a different cell type. So are EVs tissue specific, at least in regards to their rejuvenation capability? I'm not sure.
Did you happen to see a recent paper from Saul Vieta about PF4, platelet factor 4 from young plasma, that seemed to have some rejuvenating effects? And what I don't know is whether that platelet factor 4 was inside EVs or not.
Yeah, no, I don't.
Something that I find interesting is they said that they found nanog showed up in the cells.
Yeah.
It's interesting for two reasons. One, it suggests you got pluripotency, but what's a little more interesting to me is nanog showed up and you didn't get teratomas. Like, a lot of literature suggests that, if you get nanog in cells, it means you've reprogrammed them too far and you're going to get a teratoma. Like, that's kind of like a leading indicator for teratoma formation. But this suggests that perhaps things are not nearly that simple, as is always the case in biology. And in fact, nanog itself is not a leading indicator, at least by it alone, of teratoma formation, because they don't indicate that these rats had any teratoma problems or cancer problems, yet they had cells that were expressing pluripotency factors, or markers, I should say. Because if I understand correctly, we generally do not see nanog in adult cells, like, at all, right? Normal healthy adult cells, it doesn't show up.
I can't answer that clearly. I don't know if there's any conditions under which nanog might be at least transiently expressed. So I'm not sure about that. But yeah.
It does suggest that something in the extracellular vesicles it hints that something is leading to some amount of reprogramming, so to speak, which is interesting.
Yeah, well, whatever it is, it's in these EVs, and it's not in the serum, or at least not in the cellular fraction. So,
it's just the fact that you've got something that circulates, something in circulation in young animals seems to cause reprogramming or reprogramming-like things, which in itself is interesting to me. Like, it means that perhaps when you're young, this reprogramming is already happening to some extent naturally, and that's why you stay young. And maybe when you're older, those things stop happening.
Yeah. Again, I'm sure there's a lot of review papers I could look up on this, but as far as cell communication is concerned, traditionally you think about free molecules like hormones or small peptides or larger peptides circulating around. as far as particles are concerned, you think about vesicles or quasi-vesicles that carry things like lipids such as cholesterol. But there's a lot of research being done now on these like small, tiny, nanoscale particles that have a number of different factors packaged in them, which really complicates things with signaling, right? Is there a certain randomness or stochasticity to what's packaged in there, or are these things really, really finely tuned and prepackaged to the proper molar ratios of basically a cocktail of signaling factors that delivers basically a complex series of signals that kind of in like one shot, you have an integration of a whole bunch of signals that delivers a biologic message, rather than one thing that binds to a cell. Like the traditional studies is you have a receptor, you have a ligand, and you have a biologic response, right?
My guess is much closer to the latter. Like I think that people underestimate the complexity of biology and how. I don't know. Just every time I think I figured out biology's complexity, some new paper comes out and but wait, it's more complex than you thought. And so I think it's naive to think that it's just this one thing that does this one other thing. It seems in all cases it's always these 10 things do 10 things and they all talk to each other. I mean, like the hallmarks of aging, right? You see that every time you touch any one of them, all of the other ones get affected. And that's just one of the million examples of situations where it's never as simple as just one thing is talking to one thing, like ever.
Yeah, I guess the challenge here is to kind of parse out what are all the different messages in the extracellular vesicles, right? Because there's a lot of big complexity there. One layer of complexity is different EVs from different cell types and different tissue types, and then the different size range and of the EVs themselves and their whatever their components are, and then there might be a temporal component as well. I mean, EVs being released in response to different signals, by the same cell. So then you might have the same cell type releasing EVs that have different messages in response to different internal states that the cell is in, right?
Yep.
And you could just keep going on and on with that. But I mean, despite at some level, despite all of that complexity, all those layers and all those questions you ask, the fact that you get this, it's still, despite all that complexity, you can do something that's somewhat crude, right? Which is just isolate extracellular vesicles from young, neonatal cardiac tissue, inject them into an older mouse, and you have a massive improvement in cardiac function.
Yes, I'm a big fan of that. I definitely encourage people to go and try to figure out all the minutia of how these things work. But I'm definitely leaning more in the camp these days of let's try to find some broad stroke thing we can do that seems to work generally well. Like trying to understand exactly how the human body works is something that I'm not convinced we will solve in my lifetime, whereas being able to hit it with a hammer in the right way that it rejuvenates, I think, feels much more realistic. And so the releasing a bunch of extracellular vesicles from neonatal themselves or whatever feels like the hammer approach that may just work. I just looked up some random Forbes article, so take that for what it's worth, does indicate that nanog does not exist in adult cells. It is only found in pluripotent cells during early development. I'm sure they got this information from somewhere else, but at least at first blush, it does appear that nanog is not normally found in adults, and so the fact that it showed up in these mice as adults, and I mean, seems very significant to me. Like having pluripotency factors that normally don't show up in adult cells show up in adult cells. It feels like a big deal. So that alone makes me a big fan of this.
Right, and without any supposedly without any other negative consequences, but only positive consequences here. Right? Yeah. So does anybody have any further insights or thoughts? EDA?
Well, so first, I agree. The important thing here is that we have blood going to giving beautiful results for the cardiac system. But my usual question is why don't they test on real longevity? I think it's once again they killed mice or rats. I don't remember after, and they don't check for a longer possible life. But maybe I'm wrong.
Yeah, I mean the short answer I always have is that it takes a long time to do that, and it's quicker to publish this way.
Yes, I know.
That's the cynical answer. But
I even disagree. I mean, you could make a 2-time study. So you have one study, you kill 50% of the animals, and you check, and then you keep the other 50% and you wait. So for me,
they didn't mention it in discussion, but who knows? They might have a colony still going.
Yeah, for me, it's kind of crazy that so many scientists. Yeah, I said it already, but totally crazy that so many scientists are studying longevity without checking for longevity. It's not to understand.
Okay. I kind of wish that we could I could put out an announcement to all these scientists and say, if you've got rejuvenation effects in mice and your study's about to end, please just give me the mice. Don't kill them. I will buy your mice from you with your therapy applied, and I will just keep them alive in a lab until they die. Like, I will cover the cost of if you've got something that's promising.
Yeah, and it's not to keep the animals alive just without treatment. It's not so expensive. So I really, for me, my conviction is I'm afraid that these people think, okay, it will not work on the long term, so it's not necessary to check. Otherwise, it's crazy. I don't understand.
Yeah, well, I mean, lots of different labs, lots of different reasons. Space constraints probably being one of them. Time constraints.
The grad student that was taking care of the mice moved on.
Priorities shift. The grant ran out. All depends on the specific lab, right? All depends on but at least the data is published.
So, sorry to continue, but actually, okay, one new drug, it's €1 billion, and one test with mice is like, it's less about €1 million. So I mean, it's for big pharma, big drug companies, it's nothing, and keep them alive, keep 50 mice or rats alive for until they die. It's, I don't know. I think it's like €100,000 or something. It's really not expensive. Yeah, it's one. Normally you say something like $1 a day. So 1 year less than $500 per mice. So 100 mice, 50,000. So not so much money.
I mean, you should put together one of those charity commercials that say for $1 a day you could extend the longevity of a mouse and have like a little mouse that's.
You too can help this mouse survive until it dies of natural causes.
You could adopt this mouse, and for $1 a day, only for the price of a cup of coffee. Yeah. Although coffee costs a lot more than $1 these days.
that would be a fun lifespan.io, I think, one of your guys' crowdfunding things. I think you could really spin that, I think.
We can keep these experiments going for much longer, for just $1 a day.
Get some famous actor or something to stand up there, sad, holding a mouse.
And Didier, you can give the message because you're very impassioned about that, so you can deliver the narration, and maybe we can pull some money together. And
yeah, but for example, concerning blood, I was proposing to the convoys to do it. I was saying, okay, I'll pay, but they refused. So they didn't want it. They said, we know already, so it's not necessary. Okay, yeah.
Yeah, that one in particular, I am confused about the convoys. I don't know why they don't. I mean, I guess maybe we can ask Irina when she talks to us.
Yeah, so there's a potential. So looking forward, there's a potential that Irina will be joining us and leading the discussion of their latest paper that got published, which I believe is, I haven't looked at it yet. I haven't read it. I looked at it, but I haven't thoroughly read it, which is looking at a different, I believe, a different analysis of biomarkers for biologic aging. Steve is queuing that up, lining that up to see if that's going to happen. Hopefully, it will. Steve.
Yeah. It's on, man. It's on. But yeah, spoilers. But yeah, we are going to be joined by hopefully Michael and Irina Convoy next time.
Excellent.
And they developed a form of, they're calling it a barometer of aging, which is an interesting. Again, it ties in with the need for better biomarkers. So yeah, no, it's fine. It's not a secret. I'm messing about.
No, it's not a secret.
So yeah, I think it could be kind of cool. I think the rationale, based on some conversations with researchers, were not doing lifespan studies because obviously there is the cost consideration. I think sometimes they say, well, if we demonstrate rejuvenation, we can just use this standard curve for longevity, and if they're biologically, I don't know, 40, and we roll them back to say 20, we can just make an educated guess. So I think that's the rationale in a crude way behind why some people don't do it. Obviously the cost and other things.
Yeah, and time is a factor too. You got only so many personnel working at a lab and taking care of things, and then priorities shift, and then you got to take the technicians to go focus on something else, and it's.
Yeah, I mean, I don't necessarily agree with that. I think we can learn things from lifespan studies. We can even learn a little, I think, from progeric mice, but they are problematic for a variety of reasons, which I'm not going into right now. But they can actually serve as a useful sort of tool for scouting out potential therapies, and then you conduct the experiment properly in, something like Black 6. But yeah, I mean, for me, I'm much more keen on the translational side of it. So if we can demonstrate rejuvenation of, I don't know, in this case, the heart in animals, let's try and do it in humans and cut to the chase. But, maybe I'm just a little bit optimistic.
I kind of disagree, because as long as you don't know if the mice are really living long, you don't know. For example, it could be that it rejuvenates the herd, I don't know, and then the kind of metabolism is accelerating, and after that the animals die faster and not slower. So that's just one example. I don't have any idea, but I mean, for me it should be. It should be a kind of obligation for the people testing things for longevity on mice or rats to keep a few mice, at least, I would say 10 or 20 mice or rats alive with the control group also. Otherwise, it's nonsense. Yeah, and so coming back to what I said for accelerating, so if you rejuvenate, you accelerate in a way, and so it can be dangerous also. And also you can have something working on short term, but not on long term. For example, all things related to blood. It was Aubrey Dedray who was saying, okay, in my opinion, probably it's changing, but very in a very temporary way. But okay, if you can, of course, then have something used each 2 weeks or so. Okay, that's it. I don't understand. For me, it should be a totally non-brainer that you have to have, and until until I would say 10 years ago, it was a non-brainer. You had almost no scientific articles claiming that there is rejuvenation of mice or rats without having information about their longevity. That's it for me.
If it makes you feel any better, I just commissioned 3 studies. All of them will do longevity outputs. In fact, in my opinion, that's the primary output. And then we may sacrifice a few mice along the way to look at, histology early on. But the primary goal is longevity. So I generally agree with you.
Yeah. Another question I had here is that, they looked at a couple of factors, but, obviously there's a lot more hallmarks of aging to look at. And kind of one fundamental question about all of these rejuvenatory effects, even with, cellular reprogramming with OSK or OSKM, is the stability of these rejuvenatory effects, right? Meaning, how many times can you rejuvenate, or is there basically as diminishing returns? So if you Steve said organs on chips is also becoming much better, right? So just with when you cellular reprogram with OSK, and that's a question I think that we're teeing up to answer, Micah, with some collaborative studies, and same with these extracellular vesicles. Can you repeat this cycle, over a period of time and keep rejuvenating cardiac function, or does it stop after a certain point, right? That's an unknown question right now, is the stability of these rejuvenatory effects in situ, at least with adult cells.
In this study, how long did they continually reapply the treatment before they gave up or stopped?
It wasn't that long. It was they looked at 16 weeks, but I think if you look at the first.
Yeah, you're right. After 16 weeks of follow-up, animals are sacrificed.
Yeah, 16 weeks. So,
it's not nothing.
It's not nothing, but, how long can you stretch this out? And it would be interesting to see, whether that, curve of these, these improvements starts to go off, even if you, keep injecting, right? Is there, and can we learn something, from that? Is there a, and then the question is, if that does happen, which I predict it will, I can't imagine, I don't think it's going to go on forever. Then the question is, why does it taper off? Are there other, is there other, I'll say entropic changes, right? Other aging changes that happen that can't be ameliorated with EVs or OSK or whatever you're doing. And that can then point you in a direction that there might be as yet another hallmark of aging that we are missing here. That's more fundamental.
I'm sure there are many.
Right, but doing that, would OSK be, is this, if we can figure out whether or not we get cellular reprogramming happening, and then let's say we add OSK on top of this, is this additive or are they both targeting the same thing, right?
Yeah, and I wonder if partially reprogrammed cells will start emitting these EVs on their own. And so you might get, even if you don't do full systemic partial reprogramming, maybe if you can hit, 75% or 50% of the cells in the body, maybe they will then release these EVs, which then it looks like does trigger some amount of reprogramming in other cells based on that nanog expression. And so maybe this is kind of, like you said, it's a feedback loop, and you just need enough to overwhelm the default state of being old. And so if you get enough cells to be young, they'll kind of tell their neighbors, hey, we're supposed to be young with me. Mm-hmm. Or if you don't hit enough, maybe the neighbors will tell them, be old with me.
Yeah. But if you get all these changes happening, if you get telomere length being maintained, if you have which is just actually, now I'm thinking about it, wouldn't you get a substantial increase in telomere? Because these are, in rats, they tend to have very long telomeres to begin with. Anyway.
I don't know. Cellular reprogramming doesn't seem to actually lengthen telomeres, as far as I can tell. I looked into that, so I think that's a problem to be solved
Really?
as well. But the good news is mRNA vaccine technology is close to being deployed for telomerase activation. I wrote about it last week, I think. So they're teeing that up to do clinical trials for idiopathic pulmonary fibrosis and a couple of other conditions that are fibrotic in nature. Like the Blasco work shows from, I want to say like 2012, 13, they did some work on fibrosis, and the telomerase activation does seem to alleviate it. So yeah, I would like to see an OSK and telomerase activate transient telomerase activator combined. That would be very interesting.
Yeah, agreed.
So yeah, so I think we're in for some exciting times. It's like I keep saying to people, it's like, don't get necessarily excited about the latest hot thing right now, which is cellular reprogramming. Get excited about the stuff that they were talking about 10 years ago, which has now done all the stuff, and a lot of it's now teeing up, ready to potentially move into the clinic. So there's a lot of things that were being spoken about 10 years ago that are starting to come to fruition. Telomerase activators is one.
For extracellular vehicles, do we know how they're produced by the cell? Like how the cell goes about actually making them and filling them and with the right stuff, etc.?
I don't know off the top of my head. There's a number of review papers out there. I gotta dive in and educate myself on the mechanisms. I know that there's several different types. I remember reading one a while ago. They were, ones where you have vesicles within a vesicle. Other ones were basically just, you can bleb out. But I don't know the actual mechanism of where you get, packaging of the vesicles and, yeah.
I'm pretty sure that there's an article on the website somewhere in the topics that actually talks about the actual mechanisms. Like the ribosome assembles it and does this, and then it passes it to this, then it gets sent out for transport. And yeah, you are right, there's at least 3 broad types of vesicles, and they all vary in size as well. So I think it's even in that article. It's somewhere in the depths, because I remember writing it about 3 or 4 years ago. So
I think there may be a couple of pathways, one from the endoplasmic reticulum through the Golgi apparatus, and another caused by exocytosis from the lysosome. Maybe there's others, but those are...
Yeah, I mean, those are, to John's point, I mean, those are the traditional secretory pathway routes where you have vesicle transport taking place. But, as far as EVs that are used in signaling, per se, versus, other distinct, vesicles that are used to secrete various factors. I don't know. It's a good question. None of my research in the past has been focused on this particular aspect of cellular biology.
These EVs were isolated from neonatal rats or neonatal rats. Do we have the ability to generate these in a more synthetic way, such as by fully reprogramming some cells in a dish and then getting them to excrete? Like, we manufacture this, or do we literally need neonatal cells in large volumes?
There was one of the papers that we discussed that was looking at EVs that were being packaged, and I think they were packaging with expression of collagen. I think if you recall this, Steve, they had a cell-based system where they basically transfected the cells with plasmids, and they induced them with they were electroporated, and they were induced to essentially shed these EVs, and then they were able to kind of harvest them, and this was used for skin rejuvenation. I don't know if you recall that paper, Steve. So they had a setup. Where they were able to kind of manufacture EVs using this kind of small scale bioreactor.
do they? So the original. So what I'm thinking about is, as with all things in humans, immunogenicity is a problem that usually isn't in lab animals, right? Because lab animals you can get basically a clone of the lab animal, get a neonatal version of it, extract stuff from it, and then put it in an old version. And with humans, it doesn't work because you end up getting an immune response if you take out something from one human, give it to a different human, just due to the heterogeneity. And so what I'm wondering about is, it sounds like in this paper and the one you talked about, they have ways of extracting these EVs from cells. The question is can we get that first cell from an adult human and do it like a cell therapy type thing, where you take some adult cell out of Oliver, and you go through some preparation process, you extract a bunch of extracellular vesicles, but of neonatal Oliver, and then inject those extracellular vesicles into old Oliver. I think so that way you don't have the immune response.
Yeah, Steve put something in the chat. So I think I think that's a question to ask Irina. But in theory, it sounds like right. If you reprogram, if you make my cells into iPSCs and you reprogram them to age 0, then will those? What you're asking is will you get EVs that are basically completely, my protein, so there's absolutely no immunogenic response if I inject them and they're floating around in my bloodstream? I would say in theory, yeah. But in reality, I think in practice, Steve just posted Juven are working on EV therapy. Nada Youssef, ex-Comboy Lab, is CEO. So Irina might know more about this, and hopefully she and others from her lab will be able to join us next month, and we can veer off a little bit from the topic of the paper itself and kind of ask more broader questions with regards to this. I'm going to make a note of it to ask those questions. If you don't,
you're muted, Fiona.
A related thing is Immunus, which is making non-immunogenic secretome of immune progenitor cells that they're injecting, and they're now running a 1-2 phase trial on muscle atrophy. It's looking pretty good so far. It's an off-the-shelf type of product, non-immunogenic, should be
interesting,
available. Yep.
Yeah,
so,
I mean,
Hans Christian, the CEO, could probably elaborate on that.
It's definitely
anyway. Oh, sorry.
Definitely would be better if we can get a universal cell line rather than having to take Oliver cells to inject Oliver. I would much rather get a universal cell line that we can just give to everybody.
Well, I mean, there's precedent for this now with, cell-based therapies, especially with CAR T-cell therapies. I mean, people are, harvesting cells and reprogramming them and putting them back into patients. So,
yeah,
it
is prohibitively expensive at the moment, though costs will come down as we scale. I think enough companies now are on the take cells out, do some stuff, put them back in, that the cost of that process will hopefully come down just due to scale. As more and more companies need that, we're going to start seeing more and more companies develop resources into making that process cheap. So hopefully we'll see it won't be crazy expensive in the future to take all of our cells out to rejuvenate all of our.
And we could certainly envision once we characterize these EVs, you can certainly start thinking about synthetic EVs, right? Liposomal EVs with having just the right mix of factors that accomplishes the same thing. But that is in the future. And we are of course on the road there. So I don't know if anybody has any other further insights or questions, but I unfortunately have to peel off soon. Friday I have errands to run. Tuesdays I can ramble on for a lot longer, but Friday I have to actually have to take care of some other business.
Maybe we'll stick to this new Friday then. With that said.
Oh, okay. I see.
I'm messing about. I'm joking.
Hey, that's fine with me.
But yes, you are right. You've got to rush, and I'm afraid Chinese takeout has just arrived, which is definitely not healthy. But it's Friday. I know I'm a bad example, but it isn't very often. So and the weekend is here. Well, here in the UK, the weekend has already arrived. you're only 5 or so hours behind us, so I'm sure it's on its way. So hope you all have a good one. And it's been interesting. As I say, we have hopefully, well, there's no hopefully about it. Irina said she's coming, so we will have Irina Conboy, hopefully Michael as well. And I'm sure we can have a fascinating conversation. I know I've got a question.
Yeah, Micah, Friday isn't very often in the UK. They're on the metric system. It's every 10 days.
Yeah, metric. I know you guys over there have an innate distrust of the metric system. But just to confuse you, we use both here. So, yeah, it really confuses the Europeans and it really confuses the Americans a little more because we use both.
Okay, awesome.
I believe it also confuses
don't know
the
how it
British.
makes sense.
Well,
Yeah, so we use miles for distance in a car, and then we'll start talking about centimeters, meters. I don't know. I just don't get it.
Okay, well, enjoy your pint with your Royale with cheese over the weekend, and I will.
We do have pints. We are allowed to have pints.
I don't know if you got that reference to.
I do.
Okay. All right, I gotta take off, but take care, everybody. Thank you for joining me on Friday, and we'll see each other in October. So.
Yep. Thank you.
Thank you all.
Thank you.
Good to see
Take care,
everyone.
everybody.
Bye-bye.
Bye.